Okay, so structure and functions of organelles. Compartmentalization and division of labor within the cells are even more obvious with an electron microscope than with a light microscope. We will now consider the structures and function of some of the cell components in more detail. Now, division of labor, so if understand that different organelles does different work, and compartmentalization means each organelle is separated from the surrounding with a membrane. So it is more obvious, means it is more proved and seen and experimented easily by using the electron microscope, because you can see the organelles in great details. Now, nucleus. Let us know about the organelles. The nucleus, figure 1.18, is the largest cell organelle. It is surrounded by two membranes known as the nuclear envelope. The outer membrane of the nuclear envelope is continuous with the endoplasmic reticulum. Now, it is a double membrane bond organelles, and so also cell membrane is double bonded, and most of the organelles, cell membrane is double bonded, and most of the organelles are also double bonded. It means that it has two membranes, two layers of membranes, phospholipid membranes. We will look at the structure of phospholipid membrane in great details in the later chapters. You do not need to worry, this is the first chapter. The nuclear envelope has many pores called the nuclear pores. This allows and control exchange between nucleus and the cytoplasm. Examples of substances leaving the nucleus through the pores are mRNA and ribosomes for protein synthesis. Examples of substances entering through the nuclear pores are proteins to help make ribosomes, nucleotides, ATP, and some hormones such as thyroid hormone T3. Within the nucleus, so nuclear pore controls the nuclear pores helps the entry and exit of substances, and it can also control it, selective entering and selective exiting of substance. It can do it. So we will see how does that. That cell membranes, we'll basically look at it in cell membranes, and all of them are literally almost the same. Okay. Within the nucleus, the chromosomes are in a loosely coiled state known as chromatin. Except during nuclear division, chromosomes contain DNA, which is organized into functional unit called genes. So we will know that each organisms has cells which has definite number of chromosomes, but chromosomes can be seen in only when nuclear divides. Cell division, where the nucleus also divides, so that the daughter cells can have the exact number of chromosomes. But when it is not dividing, when the cell is not dividing, during the interphase, we will also look at the phases of mitosis and meiosis in the later chapters. So when they are not dividing, the chromosomes are uncoiled into chromatin fibers. The chromatin fibers contains the DNA, which contains the genes. Genes are the functional unit because each gene has a function, like gene for normal hemoglobin protein, or genes for abnormal hemoglobin protein, which is the sickle cell anemia, which causes the sickle cell anemia. So genes are they carry a function, and we'll also look at this genetic stuff in great detail in the later chapters, that there are alternative forms of genes called alleles, and genes consist of groups of nucleotides, and 3 nucleotides produces 1 amino acid, and thus we will look at them and how this amino acid joined from proteins. The proteins does the function, and like this they can control the activities of the cell. So very important. Genes controls the activities of the cell and inheritance. Thus nucleus controls the cell's activities. When a cell is about to divide, nucleus divides first, so that each new cell will have its own nucleus. Also within the nucleus, the nucleolus makes ribosome using the information in its own DNA. There's nothing to explain here. Okay, so now let us look at the function and structure of endoplasmic reticulum and ribosomes. So basically think about this. Endoplasmic reticulum makes the proteins and fats and steroids, or ribosomes, which are in the endoplasmic reticulum, which makes the proteins. Okay, let's see. When cells are first seen with the electron microscope, biologists are amazed to see so much detailed structure. The existence of much of this had not been suspected. This was particularly true of an extensive system of membranes running through the cytoplasm, which became known as the endoplasmic reticulum. The membrane form an extended system of flattened compartments called sacs spreading throughout the cell. It's like this, that if you see the structure of this endoplasmic reticulum, well, it cannot be seen clearly. Look here, the endoplasmic reticulum, they are continuous with the nucleus, like they are extended system from the nucleus. And look at this, inside this endoplasmic reticulum, I wish that there was a better image in the book, a very highly magnified one. You could have seen that there would have been sacs, like compartments. Okay, so this is the structure, and there are more structure. Let's look at them. Process can take place inside this sac separated from the cytoplasm. Good, because there are a lot of sacs. The sacs can be interconnected to form a complete system. It's like this, that this whole thing is not a sac as seen in this image. There are tiny sacs compartments, and these compartments are interconnected to form the endoplasmic reticulum. It's like this. If you think that, say for example, this is the nucleus, and this whole one sac, oh my god, what kind of drawing is this? Okay, suppose that this is the nucleus, and you think that, I don't know, maybe this whole single sac is the endoplasmic reticulum. No, now we will magnify it. Okay, we will magnify this part. Okay, let us magnify. If we magnify this part, if we magnify this part, we can see that there will be like compartments, many compartments, and they're interconnected. So many compartments interconnected that they look like one endoplasmic reticulum. But endoplasmic reticulum, many sacs together has made the endoplasmic reticulum. It's hard to draw even, it's I wish there were even better images in this book, but don't worry, you will see those images gradually when you come to later chapters or read other books. Okay, the connections have been compared to the way in which the different levels of a parking lot car are connected by ramps. The endoplasmic reticulum is continuous with the outer membrane of the nuclear envelope. Continuous means it is extended from the nucleus. You can say it is a part of a nucleus. But that is not the part of a nucleus. From the membrane it is extended out. There are two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is so called because it is covered with many tiny organelles called ribosomes. These are just visible as black dots in figure 1.18 and 1.19. At very high magnifications, they can be seen to consist of two subunits. The ribosomes consist of two subunits, a large and a small subunit. Ribosomes are the sites of protein synthesis, which are in the endoplasmic reticulum. But yes, ribosomes can be also found free in the cells, but the rough endoplasmic reticulum, it is most dense. Okay, they can be found in the cytoplasm as well as on the rough endoplasmic reticulum. They're very small, only about 25 nanometer in diameter. They're made of RNA and proteins. Proteins made by the ribosomes on the rough endoplasmic reticulum enter the sacs and move through them. So like when the proteins are made by the ribosomes on the rough endoplasmic reticulum, they enter the sacs in the endoplasmic reticulum and move through them. So yeah, that's a normal thing, nothing much to explain. The proteins are often modified in some way on their journey. small sacs called vesicles can break off from the endoplasmic reticulum, and these can join together to form a Golgi body. So proteins are modified on their journey, not the journey in between the endoplasmic reticulums and the compartments, but the journey from the production which is produced in their ribosomes and to their final birth. So yes, we will see that Golgi body has an important role in modifying the proteins produced by the endoplasmic reticulum, the rough endoplasmic reticulum. We will see that later. And then we can see that how Golgi body is formed. Small sacs called vesicles can break from the endoplasmic reticulum and can join to form the Golgi body. They form part of the secretory pathway because the proteins can be exported from the cells via the Golgi body. So Golgi body transports the protein, and then we will also see that they also processes the proteins. And Golgi body is basically formed from the sacs which break off from the endoplasmic reticulum, and they join at other end. Also some other proteins are involved sometimes. Yeah. Smooth endoplasmic reticulum, so called because it lacks ribosomes, has a completely different function. It makes lipids and steroids such as cholesterol and reproductive hormones, estrogen and testosterone. Okay, now let's see this Golgi body, Golgi apparatus or Golgi complex. Why? We'll see that why there are three names. We will see that I already told you that Golgi body are formed from the vesicles. The endoplasmic reticulum breaks down to form the Golgi body. Now there are also some associated other vesicles with this, which are Golgi vesicles. So the vesicles and the Golgi body together are called Golgi apparatus or Golgi complex. Let us see it in detail. And I've also told you the function that it processes and transports the proteins. So yeah, proteins needs processing. We'll see later after this chapter that protein has structure, primary, secondary, tertiary, quaternary, and that there are this specific structure defines a specific protein. Okay, now let's see. The Golgi body is a stack of flattened sacs. So flattened sacs which bud off from the endoplasmic reticulum. Like stack means that a lot of stack of flattened sacs. The flattened sacs which are budded off from the endoplasmic reticulum. Quite many flattened stacks stack together, like they stack together. Look at this, they are stacking one together, each sac stacked on one stack, so other stack. So stack of flattened sacs is called the Golgi body. More than one Golgi body may be present in a cell. It doesn't mean that more than one sac, it means more than one Golgi body. Stack of flattened sacs, that is a group of flattened sacs, is called a Golgi body. And this type of grouping, maybe there are many Golgi bodies. Now, the stack is constantly being formed at one end from vesicles which bud off from the endoplasmic reticulum. The vesicles budded off from the endoplasmic reticulum will form into the Golgi body. They will be processed. Now the processing by which Golgi body is formed is quite complex, which is in very much upper level books, which I will explain later, but in this book no need. And broken again at the other end to form Golgi vesicles. So Golgi bodies breaks down to form the Golgi vesicles. Okay, so the stack of sacs together with the associated vesicles is referred to as the Golgi apparatus or Golgi complex. So there are vesicles associated with the Golgi body. Now Golgi vesicles are like other things, we will see that later, but there are a lot of vesicles associated with the Golgi body which forms the Golgi apparatus or Golgi complex. Now if you want to know it in great detail, you will know it when I will explain you the higher level books. Okay. The Golgi body collects, processes and sorts molecules, particularly proteins, from the rough endoplasmic reticulum ready for transport in Golgi vesicles, either to other parts of the cell or out of the cell. Out of the cell means secretion. If it is transported out of the cell, it means secretion. The cell has secreted this protein. Okay, two examples of protein processing in Golgi body are the addition of sugars to proteins to make molecules known as glycoproteins, and the removal of first amino acid methionine from newly formed proteins to make a functioning protein. In animals, in plants, enzymes in Golgi body convert sugars to cell wall components. Golgi vesicles are also used to make lysosomes. Right, so lysosomes. Lysosomes are spherical sacs surrounded by a single membrane and have no internal structure. So basically they are a bag, a spherical bag. In biological term you say it's a sac, and it is single membrane bond. Almost all of the organelles which we have looked till now are double membraned, but lysosome is a single membraned sac, spherical sac, and having no internal structure. It does not have an internal structure. That's because the function it carries out doesn't need any internal structure. You will see the function of lysosomes. They are commonly 0.1 to 0.5 micrometer in diameter. They contain digestive enzymes which must be kept separated from the rest of the cell to prevent damage from being done. So the function of lysosomes is to carry the digestive enzymes. Why is it hydrolytic enzymes? That's because digestion is done by adding water molecules, hydrolysis. That's why we say that digestive enzymes are hydrolytic enzymes, and they are in lysosomes so that they do not digest other parts of the cells. Else, like other parts of the cell, they are also protein, carbohydrates. They may be digested, which we do not want, right? So that's why they are kept in a bag, so that they can be outside they cannot interact with the other particles and organelles and substances in the cell. But if you need it, then they will be released. Okay. Lysosomes are responsible for the breakdown, which is digestion of unwanted structures such as old organelles or even whole cells, as an mammillary gland after lactation. Okay. In white blood cells, lysosomes are used to digest bacteria. We will look at that in the chapter of immunity, immunosystem. Okay, so basically white blood cells destroy the bacteria by first ingesting the bacteria and then digesting the bacteria. Enzymes are sometimes released outside the cell, for example in the replacement of cartilage with bone during development. The head of sperm contains a special lysosome, the acrosome, for digesting a path in the ovum, egg. Very easy, nothing much to explain there. Okay, so mitochondria. Well, mitochondria is the powerhouse of a cell. It makes ATP from ADP and phosphate. What is ATP? We know that ATP is the universal energy carrier. That is, ATP carries energy, and like the we will see the structure of ATP, and that the breakdown of a phosphate from ATP converts ATP into ADP, adenosine diphosphate, and that this for this breaking of the phosphate group, energy is released. Okay, we will look at this in great detail, but when the ADP, a bond has been formed with the phosphate group and ATP is made, like that's the energy carrier, and this process of making ATP from ADP and phosphate is called respiration. Respiration is two types: aerobic respiration and anaerobic respiration. Well, the function of mitochondria is to respire both aerobically and anaerobically. So mitochondria is basically the powerhouse of a cell. We will see them in great detail in the later chapter, but this is the first chapter only. So let us see the structure of the mitochondria. The structure of the mitochondria, as seen with the electron microscope, is visible in figures. Okay, mitochondria are usually about 1 micrometer in diameter and can be various shapes, often sausage shaped in the figure 1.22. Okay, they're surrounded by two membranes, so they are double membrane, just like a like the cell membrane. Okay, they are double membraned organelle. Okay, so nothing to explain here actually. The inner of this is folded to form finger-like cisti, which project into the interior solution or matrix. Okay, now we need to see. Well, basically, we all know what's a double membrane. Double membrane means that it has two membranes, like this, say, for example. Well, this is not the exact shape, but this is the inner membrane, and there we have the outer membrane. This is the inner membrane, And let me use a different color. This is the outer membrane. Okay, so the inner membrane, look, the inner membrane is actually folded to form cystae. The inner membrane, well, let me just use the eraser from here, is actually, it has foldings like this. And oh, it's, why did I draw it so big? It's not like this. Okay, just get the idea. Okay, I'm just drawing so that you can get at least an idea of what I'm trying to say. Is folded, and there are a lot of them. Okay, these folding structures, and these folding structures are called cystae. Okay, there is no space to write here. Okay, so nothing to explain here actually. This is something you'll learn by heart. Okay. Okay, and the interior solution or matrix. So this is the interior solution or matrix. Interior solution or matrix. Okay, so yeah, there is also nothing to explain here. The space between the two membranes is called the intermembrane space. You can understand, right, which one is the intermembrane space. This here is the intermembrane space. This green colored part, this is called the intermembrane space. It is the space between the inner membrane and the outer membrane. The outer membrane contains a transport protein called porine, which forms wide aqueous channels allowing easy access of small water-soluble molecules from the surrounding cytoplasm into the intermembrane space. Yeah, we will learn that any substance, polar or like especially the polar substance, cannot like diffuse in without the help of the transfer proteins. Okay, and so, well, how are they going to be transported in? Because we need the polar substance. You need a transport protein, which we will learn in great detail in the later chapters also. So this the transport proteins are very much specific, like water needs aquaporins, and like there are a lot of types of transfer proteins, yes, for transport of these substances. Okay, so yeah, the outer membrane contains the transport protein called porins, which forms wide aqueous channels allowing easy access of small water-soluble molecules from the surrounding cytoplasm into the intermembrane space. The inner membrane is a far more selective barrier and controls precisely what ions and molecules can enter the matrix. So the inner membrane is even precise than the outer membrane. It means the inner membrane can precisely control what can enter into the cell and what can leave out of the cell. Nothing to explain here actually. Well, I don't see what you cannot understand from here, because everything's are just informations. The number of mitochondria in a cell is very variable. As they are responsible for aerobic respiration, it is not surprising that cells with a high demand for energy, such as liver and muscle cells, contain large number of mitochondria. Liver cell may contain as many as 2000 mitochondria. If you exercise regularly, your muscles will make more mitochondria. So we will learn in the later chapters that our body, any body of life, any cells, are adaptive to their environment. So if, for example, that our body or our cell needs, our body means cell definitely, well, our body is made of cells, so whatever happens inside the cells. So if the cells see that they continuously need more energy, that is if you exercise regularly, so they will start preparing more mitochondria. And the cells which has to do a lot of work, like the liver, the liver has a lot of metabolic function, so they need a lot of energy. Okay, the endergonic reactions requires a lot of energy. We will know what is an endergonic and endothermic reactions later. So the cells which needs a high demand for energy have more mitochondria, because mitochondria is the powerhouse of the cell. Okay, so function of mitochondria and the role of ATP. As we have seen, the main function of mitochondria is to carry out aerobic respiration, although they do have other functions such as synthesis of lipids. In this book, the synthesis of lipid, the process, metabolic process, is not given. You will find it in other books, which eventually I will upload explaining. So during respiration, a series of reaction takes place in which energy is released from energy-rich molecule such as sugars and fats. So respiration is not like that carbohydrate in presence of oxygen is broken to carbon dioxide and water. For this, a series of chemical reaction takes place, and the chemical reactions are quite complex, and these I will teach you in the later chapters, which is I think in chapter 12. So since this is a chapter 1, this is just for understanding. So just understand this. You do not have to worry that why do you do not know it yet. You will know it in the later chapters. Okay. Most of this energy is transferred to molecules of ATP. So definitely aerobic respiration, that is why mitochondria is there. It's needed to produce energy to do our cellular work, our work, muscular work, cellular work. Now the question is, how is the energy transferred? The energy is transferred by a molecule called ATP, Adenosine Triphosphate. It contains an adenosine group, like an adenosine molecule, and it contains 3 phosphates. When it has 2 phosphates, it is called diphosphate, adenosine diphosphate. 1 phosphate, adenosine monophosphate. So for joining the phosphates, energy needs to be inputted, and so this energy is stored in as the chemical bond potential. This is a kind of like study of chemistry, but yeah, you do need a bit of a knowledge. So, and when you break those phosphate groups from the ATP, forming an ADP, the phosphate is broken down by release of energy. Now, you may have confusion that, okay, when one phosphate group is broken, we know that for bond breaking, energy is like inserted into the system. So, but here we are saying that when the phosphate breaks from the ATP, energy is released. So what is going on around here is that for breaking the phosphate, the process of converting ADP to ATP to ADP is hydrolysis, in which molecules of water are added into the ATP, and thus a phosphate is released. Now the question is, what does it mean? What does it says? It says that when water molecule is added, the water molecule is forming bond with the ATP molecules, and we know that bond forming releases energy. So even though the phosphate group is being broken, bonds are being formed. Think about digestion. What happens in digestion? Okay, we will understand this in detail in later chapters, but this is just for realization that although the phosphate from the ATP is being broken, the bond is being formed because water molecule is forming bond with the molecule with the ATP to break the phosphate. Okay, we will look that in the chapter of digestion and you will understand it better, but this is your first chapter, so yeah, you do not need to worry. So ATP is the universal energy carrier for the cells. So like when respiration occurs and energy is formed, AMP, adenosine monophosphate, is converted to adenosine diphosphate. It is converted to adenosine triphosphate. So like, yeah, it's an energy rich molecule. That's what this. And then the ATP is very much soluble and small and easily transported, and so the energy produced in the mitochondria can be transferred to all over the organism's body, all over the organism's body cell. So yeah, that's just it. Nothing much to explain here. The reactions of respiration take place in solution in the matrix and in the inner membrane. We know what is a matrix. It is the solution in the mitochondria, and we know what is the inner membrane. Since the mitochondria is double membrane, it has one inner membrane and an outer membrane. So the inner membrane, we know that it is folded to form cysti, and so yeah, the reactions of respiration takes place in the inner membrane. We will see the metabolic process of respiration in great details in the later chapters. So no worries. The matrix contains enzymes in solution. including those of the Krebs cycle, and these supply the hydrogen and electrons to the reactions that take place in the crystal. What it says is a bit complicated for you to understand now. That's because you did not yet learn chapter 12 or saw my video of chapter 12. There the process of respiration is in detail explained, and here is actually it is not so much organized. It is basically telling that the processes of respiration occurs in the membranes and the matrix, and we know that every chemical reactions needs enzymes. So basically that's it, but you will not understand anything about respiration from these lines yet. The flow of electrons along the precisely placed electron carriers in the membranes of the cister is what provides the power to generate ATP molecules, as explained in chapter 12. You have to look at the chapter 12. We're just we'll know that in the inner membrane and in the matrix there are different structures which does each functions of the metabolic processes, and it is just telling the location of those structures. Okay, so you need to from this paragraph learn the location, not how it works, because it will be taught in detail in chapter 12. So the folding of the cister increases the efficiency of respiration because it increases the surface area available for these reactions to take place. So cister is folded. If the cister is folded, then the surface area of the for the reactions, chemical reaction to take place increases, and since the surface area of the cister is increased, huge amount of aerobic respiration can take place, and we need energy constantly. We are needing energy, so yeah, it's a good evolution. So once made ATP, I think I missed one line. Yeah, I didn't miss one line. Once made, ATP leaves the mitochondria, and as it is a small soluble molecule, it can spread rapidly to all parts of the cell where energy is needed. Its energy is released by breaking down the molecule of ADP, molecule 2 ADP, adenosine triphosphate, 3 phosphates, adenosine diphosphate, 2 phosphates. Phosphate is being broken and energy is being released, but remember, although the phosphate is being broken, bonds are being formed because it is a hydrolysis reaction, and water is bonded to release, to break the phosphate. Okay, we will see that in the chapter of digestion, and you have known a little bit from your O levels. So yeah, this is hydrolysis reaction. It means addition of water. The ADP can then be recycled into a mitochondrion for conversion back to ATP during aerobic respiration. So in this paragraph only you need to understand where the structures are present, but the reactions will be explained in the later chapters. Alright, so let us see at a theory called the Endosymbiotic Theory. Let me explain you. It's basically researched and proved that the chloroplast and mitochondria that are inside the animal or plant cell were in fact independent living bacteria, but now they have been incorporated with an eukaryotic animal cell in which both of them are both the chloroplast, which was basically a bacteria before, and mitochondria and the cell in which they now live, which is basically plant cell and animal cell, are mutually helped. So that is basically the theory, and it was also researched. Now we will just see what data was collected and how it was transferred into this information. Nothing to learn here. This is just a normal easy theory formed from research. Data was collected and then information was extracted. So that is it. Now let us look into the paragraph. In the 1960s, it was discovered that mitochondria and chloroplast contain ribosomes which are slightly smaller than those in the cytoplasm and are the same size as those found in bacteria. Okay, so see, these are the data which are actually being collected, the basic research. Okay, so the size of ribosomes is measured in S unit, which are a measure of how fast they sediment in a centrifuge. Cytoplasmic ribosomes are 80s, while those of bacteria, mitochondria and ribosomes are 70s. Very much close. These are also data which is basically basic research, but data has been collected to increase the knowledge. It was also discovered in the 1960s that mitochondrial chloroplasts contain small circular DNA molecules, also like those found in bacteria. It was later proved that mitochondria and chloroplasts are in fact ancient bacteria, which now live inside the larger cells, typically of animals and plants. This is known as Endosymbiotic Theory. Endo means inside and symbiont is an organism which lives in a mutually beneficial relationship with another organism, the chloroplast or mitochondria with the animal or plant cell. Yeah, the DNA and ribosomes of mitochondria and chloroplasts are still active and responsible for coding and synthesis of certain vital proteins, but mitochondria and chloroplasts can no longer live independently. Mitochondrial ribosomes are just visible as tiny dark orange dot in the mitochondrial matrix in figure 1.22. See, nothing to explain here. This is actually the research. We are just talking about how is it, what happened. Okay, so cell surface membrane. The cell surface membrane is extremely thin, about 7 nanometer. However, at very high magnification, at least into 100,000, it can be seen to have 3 layers described as trilaminar appearance. This consists of 2 dark lines heavily stained either side of a narrow pale interior. Yeah, the figure is here, and the membrane is partially permeable and controls exchange between the cell and its environment. Okay, so membrane structure is discussed further in chapter 4. In chapter 4 we will explain it in great details. Okay, so for now, since this is the first chapter, only a short introduction. Okay, so now microvilli, singular microvillus, are finger-like extensions of the cell surface membranes, typically of certain epithelial cells. Epithelial cells are cells covering surfaces of structures. Okay, we will look at different types of epithelial cells and different types of microvillus. Okay, later on. The greatly increase the surface area of the cell surface membrane, which is the most important useful reason for having microvilli. This is useful, for example, for absorption in the gut and for reabsorption in the proximal convoluted tubes for the kidney. These also we will look in the later chapter, but let me explain you how do they increase the surface area. Say for example, this is the surface, and like this is the surface where absorption is taking place. Okay, compare this surface to surface that has microvilli like this, finger-like structures. Okay, so right. So this structure is microvilli, finger-like extension. So definitely the surface area of this structure is more as it is folded, common sense. And I just showed you so that you can realize how microvilli increase the surface area. And increasing the surface area is really essential for few places like absorption in the gut, as we have seen in the example. We will because the more surface area, the more absorption, reabsorption can occur, and so it will be more benefited for the organism, and for the organ that has the microvilli. Okay, we will look at this in details in the later chapters. Okay, so microtubules and microtubule organizing centers. So microtubule organizing centers are basically the place where microtubules are made and broken down. And microtubules are a filamentous structure. A structure which is made up of filaments inside the cytoplasm, which carries out different functions like transport of organelles, or they form the centrioles or the spindle, and yeah, that's basically the function of the microtubules. We'll see the function of the microtubules and how they are formed and broken down in the later chapters. Microtubules are long hollow tubules found in the cytoplasm. They are very small, about 25 nanometers in diameter, together with actin filament and intermediate filaments. They make up the cytoskeleton, an essential structural component of cells which helps to determine cell shape. So cytoskeleton is basically the structural component of the cell. Now the microtubules, actin filaments and intermediate filaments together make up the cytoskeleton. So in this book, the actin filaments and intermediate filaments are not included. So we will only know about the microtubules. Okay. They also determine the cell shape. Microtubules are made up of a protein called tubulin. Tubulin has two forms, alpha tubulin and beta tubulin. Alpha and beta tubulin molecules combine to form dimers. What is a dimer? A dimer is a molecule made up of two identical compounds. Like for the molecule of microtubule, it is a dimer because it has two compounds like alpha and beta tubulin. Okay, so that's why it is a dimer. So these dimers are then joined end to end to form a long protofilaments. This is as an example of polymerization. Protofilament is basically when many microtubules join together to form a filamentous structure, like a long tubular structure. Okay. 13 protofilaments then line up alongside each other in a ring and form a cylinder with hollow center. The cylinder is the microtubule. Figure 1.24 overleaf shows the helical structure formed by neighboring alpha and beta tubulin molecules. Let's see, look. This is a dimer, alpha and beta. Look at this. They form like a round helix with nothing in the inside, hollow inside. Okay. And look at this. This is basically a structure of a centriole. Yeah, these are basically few structures. Now they just keep polymerizing until they form really long structures, long enough which are necessary by the cell. And 13 protofilaments line up together. Okay, so the dimer join to form protofilaments, and 13 protofilaments line up along each other in a ring to form a cylinder with a hollow center. The cylinder is called the microtubule. So 13 protofilaments makes up a microtubule, and polymerization of the two dimers, huge numbers of two dimers, makes up the protofilament. Okay, nothing to explain here actually, these are just data. Apart from their mechanical function of support, microtubules have a number of other functions. Secretory vesicles and other organelles and cell components can be moved along the outside surface of the microtubules, forming an intracellular transport system. I told you that microtubules help in the transport of the organelles. How do they do that? The organelles are actually moved by the outside surface of the microtubule. They are close to the microtubule, outside surface, and then the microtubule helps the organelles to move where the cell needs it to. So excretory vesicles, vesicles formed in the cell which should be excreted, are moved outside the cell surface membrane or to the cell surface membrane by the microtubules. So they form an intracellular transport system because it happens inside the cell. Membrane bound organelles are held in place by the cytoskeleton. Yes, and cytoskeleton is microtubules with actin filament and intermediate filaments. During nuclear division, the spindle used for the separation of chromatids or chromosome is made up of microtubules, and microtubules form part of the structure of centrioles. We will see in this chapter of cell division how centrioles help to separate the sister chromatids from the chromosome. Okay, we will see that. Or in that it separates the sister chromatids in the meiotic cell division, and it separates the sister chromosomes, the pair of chromosomes, in the mitotic cell division. We will see them later. The assembly of microtubules from tubulin molecule is controlled by a spatial location in cells called microtubule organizing centers. These are discussed further in the following section on microtubules. Because of their simple construction, microtubules can be formed and broken down very easily at the microtubule organizing centers according to need. Okay, now centrioles and centrosomes. The external resolution of the electron microscope reveals that just outside the nucleus of animal cells, there are really two centrioles and not one, as such is appeared under the light microscope. So under the light microscope, we saw that a cell contains only one centriole. But when we used a better microscope, the electron microscope having the ability to show more detailed structure of the cell, we saw that there are actually two centrioles. Okay, they lie close together and at right angles to each other in a region known as the centrosome. So the region which contains these two centrioles that lie along each other at right angles is called centrosome. What does it mean that at right angles? Well, it basically means that if this is centriole number 1, then this is centriole number 2. There you go. This is what it means. 1, 2. Oh. Anyways, you'll understand, right? This is what it means, that they lie at 90 degrees. Right angle means 90 degrees. Nothing much to explain. These are just solid information. Centrioles and the centrosome are absent from most plant cells. Okay. A centriole is a hollow cylinder about 500 nanometer long, formed from a ring of short microtubules. Each centriole contains 9 triplets of microtubules. These are solid information here, nothing to explain. The function of these centrioles remains a mystery. Until recently, it was believed that they acted as the MTOs, which is microtubule organizing centers, for the assembly of the microtubules that make up the spindle during nuclear division. It was believed. Okay, so yeah, in the nuclear division, we will learn it in great details in chapter 5. It is now known that this is done by centrosome but does not involve centrioles. So there is a room for research around here to understand what is really happening. We need to reveal like the secrets of life, right? So centrioles found at the bases of cilia and flagella, where they are known as basal bodies, do not act as MTOs. So basically centrioles are found at the bases of cilia and flagella, but these are basically present in the when we breathe, the oxygen goes to our mouth into our lungs through the tract path through which the oxygen passes. So in that path there are cilia, so that cough or other dust particles from the lungs can be swept up. Also the bacteria, we will learn later, contain cilia and flagella, whose beating movement helps in their movement. Okay, we will learn this in detail in the later chapters. And flagella, where they are known as basal bodies, do not act as MTOs. The microtubules that extend from the basal bodies into the cilia and flagella are essential for the beating movement of these organelles. Okay, we will learn this in the chapter of lungs and also in other chapters where we have to see how the bacteria moves. Okay, so up until now we just have to know that what does centrioles do. Okay.